All right. Well, welcome everybody. Welcome to Seward Park Audubon Center Science, Nature, and a Biscuit program. We're happy to have you back here as we kind of get started with a new season of discovery and learning. Science, Nature, and a Biscuit is our program when we present some wonderful people out there doing science. That is the place where science and nature meet, and they have a great body of work that they want to share with our audience. And we're so happy to do that. Unfortunately, we don't have biscuits this time. Don't let that stop you. Go ahead and make your own biscuits. And a little tip for making biscuits for me, you have to add butter to it, freeze the butter, and then use a cheese grater. That way you get the really small pieces of butter and it's really easy to mix that way. So anyway, we want to thank you all for coming out tonight. Thank you for supporting what we do. And I'm going to turn it over to one of our esteemed volunteers. Twee Lu is going to be here. She's going to be the host and moderator. And so I'm going to turn it over to her and you can watch me disappear.
Hi, everybody. Thank you for being here on this lovely evening, wherever you are. My name is Twee Lu. I'm a student at the University of Washington, and I'm a volunteer for the Seward Park Audubon Center. This program is being recorded and will be available on the Seward Park Audubon Center website for the next 30 days. We will be fielding questions at the end of Lila's presentation, but you may use the Zoom chat feature. It should be at the bottom there to post your questions or comments at any time during the presentation. And now I would like to formally introduce to you our speaker for the night. Lila Westreich is a PhD candidate in the Tobin Disturbance Ecology Lab at the University of Washington, studying the foraging behavior of native bees across a variety of flowering landscapes in Western Washington. Lila received a Bachelor of Science in Plant Breeding and Genetics from the University of Minnesota. And now they are currently working to identify pollen and microorganisms collected from native mason bees using next-generation sequencing, a novel genetic analysis technology in the study of native bees. So please join me in welcoming Lila to our Seward Park Audubon community.
Thank you, Twee. And Joey, I'll let you go through the next steps, or should I take us through to the land acknowledgement as well?
You can just go ahead and hit start.
Cool. All right, so I'll hop forward here to this nice jump to my big screen. So again, thank you to Twee and to Joey for hosting me. I really enjoyed when I first gave a talk. It was in person, which was very nice, and I got my own biscuit and everything as part of the series, I believe last summer, the summer before, and it's kind of cool to come back and give an update because now I actually have some data to show you from Seward Park specifically, but I'm going to talk a little bit about my work, give some background on the bees that I study and the bees in our environment, and then take you through what my study looks like, and then some of the data that I got from 2018. So just a really small piece of what we're working on, part of my dissertation work for my PhD. So, let me close up some things here. Zoom is always a funny way to give a presentation, but I'm really glad that everyone could join. I'm actually in Minnesota, so I'm in Duluth, Minnesota, giving this presentation a little later than maybe some of you are viewing it on the West Coast, but we'll start out with honeybees. So hopefully you all are at least a little bit familiar with honeybees. They're probably the most well-known and maybe even the most loved bee currently. They're wonderful pollinators. They provide about a third of the world's agricultural pollination. As part of a lot of grants that we submit, we talk about honeybees because they're a huge contributor to a lot of our food sources, and they contribute about a couple $2 billion to our agricultural system. Without them, we would not have the fruits and vegetables that we have on our plates today, but they're also a non-native species. So they are domesticated. We bring them in. The bee that we use for pollination services in the United States is Apis mellifera, shown here. The European honeybee, which we purchase queens from Europe, bring them to the United States, and they grow up their colonies in these hives that we transport around the world, or around the country, excuse me. So in Washington, honeybees are responsible for pollinating our apples as well as a lot of cherries. In California, they truck them there for almond pollination. They're actually using a lot of native bees for almond pollination now. I'm happy to talk more about that, but honeybees are predominantly used for that work. And then they truck them through the other states to do things like citrus fruits and blueberries and all kinds of foods that we eat every day. So they are wonderful, but they are non-native, and they do carry diseases and parasites that can actually transfer from colonies of honeybees to our native species as well. So they do pose a lot of problems, and the issues that are facing honeybees are also facing our native bees. The same levels of decline and concern that we see around honeybees are actually affecting our native bees as well. And I actually personally believe that our native bees are a lot more interesting, just due to their, in part, their beauty. As you can see from all these pictures, they're absolutely gorgeous. And I think they're a lot more interesting. Our native species are really diverse. In the United States, we have about 4,500 species of native bees that have been identified so far. In Washington State, there are about 600 that have been identified, and more are being found all the time. We don't know all the species of native bees yet, but the majority of these bees are really solitary. So a honeybee colony lives in a hive, there's a queen. Not all the females are fertile, they're not able to have offspring, but our native bees that are solitary, all females are fertile, and it's kind of like a single mother responsible for everything for her children. And so they actually live in very different locations. They nest in trees, in the ground, even in the siding of your house, you might have seen some of these bees. And of all of these species, the one that I focus on, sadly, not that one in the upper left-hand corner, which is my absolute favorite native bee. It's considered the teddy bear bee is its nickname. It's a Xylocopa species. Those are found in Northern California, so sadly not in Washington, but we do have this wonderful bee in Washington, which is the Osmia lignaria, or the blue orchard bee, blue orchard mason bee. They are assimilated native to Western Washington, which means that their range at one point was much smaller, but has expanded into Western Washington. They can actually be found anywhere from southern British Columbia in Canada down through northern California. And they're actually used for pollination all over. They use them for pollination of almond trees in California. A lot of people in Washington use them in gardens. You can buy them. They're available from local breeders. And these are all aspects that make them really easy to use for my research. So as part of my research, I have to understand what the native bees are doing. And the best way to do that is usually to either capture bees off of the landscape or provide your own bees to study. And I actually purchase these bees from a local breeder and use them for my studies, using them as kind of a proxy for a native species. And so these guys are really unique. This is a male shown here. They have that kind of yellow mustache over the front of their face. And excuse me if there's any traffic out the window, I am, Zoom problems. So they are a solitary species. They nest usually in holes in trees or stems found naturally occurring, as well as the siding of houses. They love the siding of houses. And they nest well in a box design that I'll show you a little bit later on. They emerge really, really early in the spring. The males emerge first and then the females. And they are very short-lived. So I'll move you on to a picture of the female. They only live about 4 to 6 weeks, and in Seattle, they emerge around March. Western Washington, kind of anywhere from March to April is their emergence time, and they live through about the middle of June. So their time is done for now. They may still be flying a little bit if they were a little off on that timeline, but all those features make them a really good bee for studying any changes to phenologic bloom time. So if in the future we see blooms happening at different times of the year due to climate change, these bees will be the most affected because they come out earliest in the spring. And the way that they nest also makes them really, really easy to study because I can move them all over the place. They nest in these nice human-made structures, so I can steal their pollen, which you can see some pollen in the back of her thorax there, and I can use it for my research. So I'm going to zoom out a little bit and talk about Seattle as a whole and kind of the plant variation that we're seeing here before I zoom back in on my experiments, because I think it's really important to understand that Seattle as a study site is a very unique place. So Seattle, if you're familiar, it's nicknamed the Emerald City for all of its immense green space, a lot of public park systems. They've won many awards for it. But those public parks are not necessarily all the same. There's a lot of variability in what's growing and what's available, especially when you're looking at it from a pollinator lens. So, for example, this is Magnuson Park on the eastern side, just north of the university. It's huge. To anyone familiar with Seattle, it's very well known. It's one of my favorite parks for hanging out in with my friends, but if you're looking at this park from the air, you can really see why it's not necessarily a pollinator-friendly park. So yes, it's a very big green space, but you can see a lot of that space is full of areas that plants really can't grow that would benefit pollinators. So we're seeing tennis courts, baseball diamonds. There's even a soccer field up there that I actually just noticed looking at this picture now. And a lot of grassy areas where it would be great to take your kids and hang out and have a picnic, but pollinators really aren't going to be able to forage for food there or nest there due to all the human activity. Even a lot of this park is parking lots. So even though it's really big and really immense, and there are pollinator-friendly plants within it, as I'll talk about a little later, because I do have sites at Magnuson Park, it is a really different park than a place like Discovery Park. And I don't have a good picture of Seward Park, or I would definitely include it here, but you could easily swap a picture of Seward Park in for this picture of Discovery Park. So this is another huge space, huge green space, filled with flowering plants, flowering trees, a lot of really, really good nesting sites for bees. It's kept very wild and native. You know, there's a lot of work that goes into keeping these areas free of invasive plants or even non-native plants. And same with Seward Park. A lot less development has happened in these areas. And so it's really a very pollinator-friendly place. So you're seeing a really big difference between a lot of the places in Seattle that you might consider a green space when you're looking at it from a pollinator side. And then you also have to consider the plants that are in each of these green spaces. So these pictures are all from me, but and they're not necessarily taken at all of our park systems, but you can find all these plants within a lot of my study sites. So we've got 3 different kind of general classifications that I threw in here on the bottom. You can see non-native or invasive plants, ornamental plants, and native plants. And you're going to get really different pollinator friendly plants even within these general groups. So for example, ornamental plants, a lot of plants are bred for show. You know, we breed plants to make beautiful colors and to plant them in our lawns or specific shapes and sizes. But a lot of times when humans are breeding a plant, they're accidentally kind of breeding out the characteristics that make them really good for pollinators. So we may breed a flower so much, like a tulip, that it doesn't necessarily produce nectar or pollen that is useful for pollinators. Or when you're breeding a plant, you're not breeding a plant for its nutritional value and pollen, you're breeding it for show. So we kind of lose a lot of characteristics that make plants really good for pollinators when we're breeding them like that. And then in terms of invasive plants, some invasive plants do provide a lot of benefit to our pollinators. I think Himalayan blackberry is one that it's not technically labeled as an invasive, but it is a non-native and very invasive-like plant in Western Washington and in Seattle that provides a lot of beneficial pollen to pollinators. But something like Scotch broom on the top left there, which is a highly invasive plant and should be eradicated whenever you see it, according to our state guidelines, it does provide some beneficial pollen, and it is visited by bumblebees who can buzz pollinate the blossoms, but the bees that I use cannot do that. They can't even get into those blossoms, let alone obtain pollen from them. So you also have willow on the bottom left, which provides actually ample pollen. It's really highly visited by honeybees and by my bees, which I'll show you. But it's not a native plant. A lot of them are an Asian species or Eurasian species, and willow kind of interbreeds so quickly that it's hard to distinguish which was the native. You know, next year it could be a mix of native and non-native. So it's a difficult situation. And then native plants, of course, they provide pollen and nectar, but in the face of all of this diversity and variability, what are the pollinators really choosing on the landscape? So then there's one more factor to bring in, which is climate change. I am not going to talk too much about the impact of climate change on pollinators during this talk. There's so many pieces of this talk that could be a whole nother presentation, but the climate change is having an impact. So what we're seeing, and I think we're especially seeing this in Western Washington, is spring is coming earlier. Plants are blooming earlier. The way that plants need certain characteristics to bloom is very different from what our bees need to emerge. If a plant comes early, it doesn't mean a bee is going to emerge early. So if we get something blooming in February and the bees aren't ready to come out yet, they're not going to come out just because this thing is blooming. They're going to wait until they're ready, come out, and whatever's on the landscape is what they have available to them. And so this is a worldwide thing. It's not just happening in Seattle or in the United States, of course, and it definitely has a huge impact on how our pollinators are behaving in different locations. And to illustrate that, these are all pictures that I took in, let's see, I think it was the first 2 weeks of February were all of these pictures, and that kind of floored me. Actually, I wasn't even thinking about the phenological bloom time of all these plants and how weird it was. I just kept thinking, wow, there's so many flowers outside. You know, these were all outside my house, which is in Capitol Hill. So it was really kind of amazing to see this. And it was kind of scary because my bees were nowhere near ready to come out. And a lot of them would have visited some of these plants. So just something to keep in mind and something to keep track of. And it's something that we study within my lab, but not something I'll talk about too much in this talk today, just a threat to our pollinators. So to zoom in on the pollen a little bit, because this will be important down the road for my results, pollen is, of course, what bees eat. It's their main source of food. It contains the same things that are in human food, amino acids, lipids, or fats, and proteins. And these are all needed for growth and development of the bee, the same way that you need them for growth and development. The lipids and protein have been kind of the hard-hitting factor that a lot of scientists have focused on recently. So not only do bees need protein and lipids, they need them in very specific ratios, which I'll talk more about on my next slide. But it's important to note that this nutrient content varies pretty wildly by plant species and even within genera and families. And to show that, this is a figure from a paper by Dr. Anthony Votto, who is a wonderful researcher. He's a postdoc. I believe he's at the University of Nevada in Reno right now. I could be wrong. I actually just emailed him the other day. He's a wonderful researcher who studies the nutrient content of these pollinator plants. And so he's trying to understand what's most important within the pollen in terms of promoting growth and development within the bees. And so as you can see here, I'm going to move myself out of the way a little bit so I can see what I'm talking about too. But as you can see, these are the pollen, protein, and lipid ratios of these plant families. So along the left-hand side of this figure, you have all of these plant families. If you recall, family, genus, species is kind of the breakdown of plants. So this is a wide angle of all these big groups of plants that fall within the same family. And what we see is there's a really wide variety of nutrient value, which is what that PL stands for. And on the right, I kind of described it a little bit so it's a little clearer, but essentially there's really, really high nutrient quality at those top plants and the top family, and then really, really low at that Liliaceae, that bottom family. And this actually puts them in order, but there is a huge variety even within some of these families. And so what they did is they averaged all of these nutrient levels so that they had a relative breakdown of what was happening within the nutrients within the families. And then the really interesting thing that he did is he pulled out the bees as well. So these are an analysis of what is required for the development of these 3 bees. And he just used these as examples. We have an eastern bumblebee up at the top, a mason bee species in the middle there, and the European honeybee down at the bottom. And what's really, really interesting about this figure is European honeybees don't require nearly as much, nearly as high of a nutrient content to their pollen to survive as these native bees, mason bees and bumblebees. So if we're going to create a conservation area, you know, we're going to plant a bunch of pollinator-friendly plants, and we think, oh, well, we know what honeybees need and we know how well they do on all these plants. So we're just going to plant the exact same plants in this pollinator strip. That's not necessarily good enough for our native species. And I think European honeybees have been used as kind of a gold standard for bees for a long time because we really didn't know there was so much information, so many differences with the native bees themselves. And so this was really interesting, and this kind of formed a lot of my research questions as well. They'd been forming a long time ago, but this solidified that we really needed to look at the nutrient content for solitary bees and for native bees in order to create the best landscape to promote these species. So just to wrap that up, I know it's a lot. The basic needs for these bees are flowering plants. For those that emerge really early in the spring, there's obviously many species that work during kind of the normal pollination months in the summer. And then a lot of them survive all the way through to the fall. So we're going to need flowering plants that entire time for them to be fully saturated with the pollen that they need. And in terms of pollen, they need these nutrients in pollen for development. These high protein lipid ratios within the pollen are what historically in the research has been attributed to success for the bees. So all the papers that have been coming out say that high protein lipid ratios mean great development for the bees, and low protein lipid ratios mean really poor development in the bees. And then, of course, I didn't talk too much about this, but they need a suitable habitat. They need loose soil or piles of leaves that are left untouched. There's all these things that you can do to your yard, obviously, to make it a little more bee-friendly for nesting, but that's something that we lose in developed areas as well. So to talk a little bit about my study, we've made it through the intro. Now we're talking about my study sites from 2018. So I have been in grad school for quite a while. I have many more years that I've done this work, but 2018 was the year that we have kind of the most cohesive look at these sites that I've shown on this map in pink. And so I'm going to talk mostly about 2018. I'll talk at the end about what I'm doing with these sites and many others that we've established within the city of Seattle. But for now, we'll focus on 2018, which is, as you can see, the kind of bigger park systems, the Magnuson Park on the right-hand upper side there. I know not everyone's from Seattle. The left-hand side of this map is Discovery Park, and then down at the very bottom right, of course, is Seward Park. You can see my dots there, and then that clump kind of in the middle, I'm not sure if you can see my mouse, but this clump in the middle here is the Union Bay Natural Area and Center for Urban Horticulture, as well as the University of Washington Farm. So these are kind of an interesting mix of restored sites, urban sites, and I'll talk a little bit about those once we get a little further on. But a lot of what I'm trying to figure out with my work is this analogy. The best analogy that I've come up with is essentially like a grocery store versus a gas station. So in a grocery store, when you go to the grocery store for the week, you get, you know, grains and vegetables, a lot of vegetables, maybe a lot of protein if you've got kids, or just for yourself, maybe a chicken or fish. You might get maybe a bag of chips, right? Or a candy bar as like a snack, a fun treat at the store. And that'll sustain you for the week and you'll do really well. But if you end up going to a gas station and you're forced to get all of your food for the week at a gas station, you will probably end up surviving on sugar and chips, right? Pop and sugar and lots and lots and lots of kind of junk food, fast food. And so what is that going to mean for your development? You know, you're obviously not going to do as well if you're eating sugar and chips. Sure, they'll get you through, but they won't necessarily be the best thing for you, for your health. So that's what we're asking. If we put these bees in one site that has like all of these really great pollinator-friendly plants, what are they doing? Where are they going? Versus another site where there are very few pollinator-friendly plants. What are they doing there? What are the choices that they're making based on the landscape that they're in? And how do those affect how well the bees do? So that's kind of the background of my experiment, and this is exactly what it looks like in the field. So This is the box design. At each of those pink triangles, we placed 1 or 2 of these boxes, probably mostly 2. As you can see, it's a really simple design. We made these in my advisor's garage, so it's nothing fancy. We use pretty cheap materials. It's PVC pipe, so it's you can't see it, but it's about a foot-long PVC pipe that's been beveled at one end to provide some rain protection. And then inside are paper tubes. So these bees, as I mentioned before, they nest really well in human-made materials, and they love paper. It's really, it breathes a little bit. Plastic is really tough for them because it doesn't breathe. And the paper does mold in Seattle, which is an issue I run into, but usually I'm so much in the field pulling them in and out and throwing them in the freezer that we don't run into that issue quite as much. But they'll fly out of that box, so that tube on the bottom there with a single hole drilled into it, that's what we call an emergence tube. So I place the bees that I get from a breeder into that emergence tube before they're ready to wake up. They're still asleep for the winter. I place them in the field, they wake up, they come out of that hole, they look up, and there's a beautiful home waiting for them. And they go up and they fill that home with, they'll go out and collect pollen, put it all over their bodies, go back to one of those holes, crawl all the way to the back, dump all that pollen out and form kind of a little perfect ball. And I'll show you that in the next slide. And then they'll lay their egg on top of the ball and then hole up the section of that nesting tube with mud. So the mud is what gives them their name, mason bees, because they use mud to build things. But they, you can tell that each of these tubes is capped because of that. If you can see it, that capped of mud that's kind of on the end of the nesting tube. And again, if you can see my mouse, I'm kind of circling it. So this one picture on the right is a bee working. There's also a giant wasp, if you can see it, that's about to come out of that box that scared the living daylights out of me. But I managed to take a picture of it, which is pretty cool. So what we do is I'll pull all of these tubes out and they'll look like this. And we'll pull the pollen from the tube and use it for our genetic analysis. So this is a really, really successful tube. A lot of them don't look like this. This is like, you can see there are tons of offspring that fill up that tube. And you can see the cap in between, the little kind of green note of a cap. The bees sometimes pick up a little grass along with the mud that they collect. You can see all those caps separating each of them. And on the top right, you can actually see a pile of those mud caps that I pulled out from probably a previous tube. And on the bottom left, you can also see the larvae. So you can see these little larvae that are, I've halted their development. We have to in order to get the pollen, but there are a lot of larvae and a lot of successful bees within this tube. I would call this a really successful site if this were indicative of all of the tubes within that box. And we pull that pollen so that it looks like this. And in 2018, we did mixed samples. So maybe one of each of these is from a different site. And we mix them all together by site and kind of homogenize the pollen so that we had a good sample, a representative sample. And this is kind of poor science because I'm not showing you the labels on any of these tubes. But trust me that they are likely all labeled with the correct site and the correct date and all of that. So we take that pollen and we use it for genetic analysis. And I'm not going to go too deep into genetic sequencing. This is a really simplified figure. It's probably too simple for a lot of you, but I'll give you kind of a general breakdown of how the process works. It's used all over biology these days and ecology. It's become cheaper to do genetic sequencing than it is to do the old method of identification of pollen, which is you buy a really, really, really fancy electron scanning microscope and you look at pictures of pollen that are blown up. You might have seen those coffee table books with giant, beautiful pictures of pollen grains. But that takes a lot of skill as well. So you have to be able to match those pictures to a reference library. And there are experts who've studied for like 20 years to do that. And basically when I started my PhD, I thought, okay, I could either spend a couple years learning how to do that, or I could start right now and use genetic sequencing. So I took the easy way out. But the basic gist of it is that you take plant material, whether that's, or what I do at least, you take plant material, whether that's like a piece of a leaf or a twig, as I've shown here, a willow twig, or this vial is representing the pollen picture I just showed you, that vial of pollen. You grind it up so that the cell wall is broken down and the genetic material inside can emerge. and then you extract the DNA. So as you may remember from biology class in high school, DNA is in everything. It's in everything living, and we are able to break cells apart and pull the DNA out in order to read it in our genetic sequencing software. And so I do that in my lab. I pull all the DNA out. It's kind of a long, hard process that I've gotten quite good at. And then we submit that DNA as a sample to a sequencing group. And the sequencing group essentially takes the biologic material, puts it into a machine, which breaks it down and builds it up, and the output of the machine is this long, long list of A's, T's, C's, and G's. So if you may remember, the A, C, T, G bases are what make up the DNA strand. And so what the machine is doing is it's reading a little section of DNA, the section that I tell it to read, because if we read the whole thing, it would be billions of base pairs long. Don't worry about it. We read this tiny little sequence on the DNA strand, and it gives us this long output of letters. And so what I get back from the sequencer is essentially many, many, many thousands of megabytes of letters. And what we can do with these letters is we use a bioinformatic process where we plug these into what is essentially like a Google for genetic sequencing called GenBank. We plug in the sequence that matches that read, and what the Google kind of outputs is, okay, this is a 99.999% match to a willow species. And the only way we'd really get 100% match, 100% match is hard. 99.999 repeating is perfect. That's like considered perfection for us. But the only real way to get 100 is to sequence the entire genome of the plant, which for plants is enormous. And I don't have the money to do that. So we stick with the 99.9, and that's accepted in the literature. So we essentially do this with every single site. And of course, not all of the pollen is gonna match willow species. We're gonna get all kinds of plants in that output. So hopefully this makes genetic sequencing a little clearer, but it is kind of a wondrous process where you do a couple things in the lab, you send it off, and all of a sudden you get data, really, really cool data where you can understand where the bees are going and what they're doing, and a relative understanding of how often they're going to one plant over another. So now we're going into the really heavy data stuff. So take a deep breath, and I'll explain it all through for you. So this is Seward Park. We're going to start with Seward Park. We'll kind of zoom out from there. So this is a graphic, real simple. On the left-hand side are the genera. So we are able to get, in 2018, we were able to get all of the plants identified down to genus, but not to species. So this may be a list of only 22 genera, but we actually have many, many, many species within this. So just keep that in mind as well. And on the x-axis down at the bottom there is the percentage of the pollen sample that we submitted, which was a representative subsample of the enormous amounts of pollen that we got from each of these sites, and how much was identified back to each individual plant genus. And this is shown for, so I had two sites at Seward. I call them Seward North and Seward South. They're really probably more like, they're just barely north and south of each other, but it was my best, it was like 4 years ago, so it was what I went with at the time for classification. But these sites are actually within 300 meters or so of one another, so probably a little bit less than that. And these bees fly more than 600 meters to find food. And so bees we placed at Seward North may have gone to Seward South or vice versa. Just they were flying along, they saw the nesting site, they thought, hey, this makes a better home, they nested there. So I consider these kind of one location, one data point within my data, but we looked at them, we analyzed them separately because we had them separated within all of our data that we submitted. So, but it still, I think, is interesting to see that you can see that Seward North and Seward South, the bees did different things. The only thing they really did similar was this rubus, the longest, it was about 45% of the pollen sample that we submitted was attributed back to rubus. Rubus is berries, and I'll talk more about this. I'll kind of break this down a little more, so you don't have to totally understand this figure to understand the whole talk, but rubus is could be any berry, a thimbleberry or salmonberry or even a Himalayan blackberry. So they clearly liked rubus, any of the berries on both of those sites, regardless of what else they were visiting. So then I know this is a lot, so let's break it down a little bit into this nice, understandable figure. And what I did here is I threw in the genera, kind of the common name as well as the genera. Mock orange, I didn't throw it in there. It's Philadelphus is the mock orange genus. But we have the top 3 visited plants within our pollen sample. So we have wisteria, the rubus or berries, and they're all represented down at the bottom as a reference picture, because I know a lot of people really don't bother with scientific names. We kind of have to, but it's not necessary, of course. And on the right-hand side is the mock orange. And so I threw in which plant family they all came from. The reason I did that, that's second line in the table. The reason that I did that is because that first paper with Anthony Votto that broke down the relative nutritional quality of each plant family, that was really interesting to look back on once I got this data back. So I took those families and I took the nutritional quality and I threw it in here. So wisteria, which is non-native, definitely non-native, was visited about a third within the pollen sample. So about a third of that pollen sample was made up by wisteria. And wisteria is from the Fabaceae family, and Fabaceae is like the legume family, and it has this really, really nutritional pollen. It's considered like the best possible thing you could feed to your bees. And a lot of bumblebees are seen visiting a lot of legume plants, even a lot of like small kind of backyard weedy plants are legumes, and we'll see bees visiting those all the time. And those are usually within the Fabaceae family. So really, really, really good for bees. We know that for sure. In the center row, they're the rubus or berry. They're in the Rosaceae family. There's a lot of plants in the Rosaceae family. Some of them are highly bred. I would assume these bees are visiting the ones that aren't kind of ornamental plants. These are the salmonberry, the thimbleberry, all of those. Those have a medium nutritional quality. They're not the best, they're not the worst, they're pretty good. And that was about almost half of the pollen in our samples was from those berry plants, which was really interesting. And then mock orange, which is Hydrangeaceae, which not a lot of nutritional work has been done on Hydrangeaceae. It's actually not in that Votto paper at all, likely because it's not considered a pollinator plant. It's not something you would necessarily put in your pollinator garden to attract bees or to help bees. It just doesn't come to mind first. And I actually wasn't really familiar with it until I started my studies. And that was about 25% of our pollen in the sample. So a fourth of the time the bees seem to be visiting this mock orange. And I put unknown for the nutritional quality because it wasn't referenced in the Votto paper that I talked about earlier. And I really am going to have to dig to find out more information on it. And so what stuck out to me here is that we have a plant, wisteria, that has really highly nutritious pollen, but the bees aren't visiting that plant as much as they're visiting ones with lower nutritional pollen. So that tells me that maybe the nutrient quality is not necessarily what's drawing them in, which adds some evidence against this theory that the bees are only going to go to the best possible thing for them. So now this is a lot. I threw you into it because I trusted you, but this is kind of a lot. So bear with me. On the left-hand side, we have our our genera that were visited. So all of the plants are on the left side. All of the right side is the sites. So as you can probably see, I talked about the UW Farm, the Center for Urban Horticulture, the Union Bay Natural Area. Magnuson Park is there, Discovery Park, and all the way at the bottom is Seward Park. So this is a plant-pollinator interaction network, but it's only with one species of pollinator, the Osmia lignaria that I work with. And all of my sites in Seattle on one side and all of the plants that they were visiting on the other. And I pulled out these really big names, Salix and Rubus, and I'll give you a break here and throw in a picture of those. So you don't have to focus on the scientific name, you can focus on the picture, it helps me a lot. We have willow, this is Seattle-wide, by the way. This is not just Seward Park, of course. We have willow and rubus. I put a picture of a Himalayan blackberry blossom, but again, could be any berry, any of our native Pacific Northwest berries or the Himalayan blackberry or trailing blackberry. And this was for May in 2018. And so what we're seeing here, and I outlined that pink box to make it a little easier for you to focus on the park we're in right now for our talk. This allows you to see the diversity of what the bees are doing and the relative amount that they're visiting one type of genus over another. So Salix has that giant black bar on the left side. It's really, really wide, and you can see there's a lot coming out of it. which I can tell you from looking really intimately at that data, the Union Bay Natural Area, or UBNA as we affectionately call it, on upward, those sites are very close to a big area that's covered in willow plants. And it looks like those bees visited almost entirely willow. A majority of what they were visiting was willow. They were visiting maybe 10, 15 plants total. Some of them only visited seven plants total, 7 types of plant total. So down on the bottom right, in comparison, we have Seward, and you can see all of those lines going out from Seward Park, tons of lines, way more than up top. And the Seward bees are not visiting willow the same rate. There is willow around, not nearly as much, so that does make sense, but there's a lot more variability in what they're doing at Seward Park. And so this just tells us about the general trends and patterns. It doesn't tell us a whole lot about what matters to the bees, but it definitely shows us what's going on in the city of Seattle. And so to give you some wrap up here, so you don't have to think too much about that really, really busy figure, the willow is really, really dominant across a majority of the Seattle sites, though not the Seward Park sites. And Seward was really unique in that, and I'm really excited for 2019 and 2020's data to come back so we can understand even more about what's going on there. The berries, these flowering berries, they were definitely favored by the bees. You can see there's that's a really big black bar in front of the berries there, but it's not necessarily as much as the willow, and it doesn't seem to be favored nearly as often, but it is visited more often than a lot of those other genera that are listed there. And there's tons of variation, just so much variation within the city. So this is really exciting stuff, but unfortunately it just brings up more questions than answers, which I think good science brings up more questions than answers. But to wrap that up, in all of the samples from Seattle, so all the Seattle foraging preferences that we're seeing here, about 1/4 of the visitation was to willow, which makes sense because some of the sites visited willow almost exclusively and some didn't at all. The rubus or berries, we're seeing another 1/4. So in total, willow and these rubus species added up to about half of the data. And then the mock orange was still visited at 10% across all of Seattle. So it was visited 1/4 of the time in Seward and 10% across the entire city. So it's clearly of interest to the bees. And again, I threw the nutritional quality on there. So far for willow, it's not really considered a highly nutritious plant for pollinators in comparison to something like the bean family that I talked about, or rubus, which has a little bit higher nutritional quality. So again, this points to the bees maybe aren't choosing something because of nutritional quality. They might have a completely different reason for foraging the way they do. So in conclusion here, kind of wrapping it up before I tell you what I'm doing next, the genetic analysis tool is really powerful and great. It works really well. We get really good data that makes sense from being on the landscape. When I've been in these sites, I've seen a lot of willow in places where the bees were visiting willow, and a lot of berry species in places where bees are visiting berries. It works really well for understanding their behavior. And certain genera are definitely playing a much larger role in their foraging patterns than was kind of previously assumed. I wouldn't have necessarily assumed that willow had such a big impact on the bees when I first moved to Seattle. And those foraging patterns don't seem to be very clearly linked to the nutritional content, at least for the 2018 data, which could be evidence against this theory that bees are choosing plants on the landscape because of their nutrient value. And so again, brings up tons more questions than answers. Really doesn't solve a whole lot, but it points us in the right direction for the next steps. And so just to bring you in on what I'm doing next, these are 2019 and 2020 sites on the right on that map of Seattle and kind of the greater King County area. We've got sites on Vashon, a lot more sites within the city to show more variability in what the bees are doing. We also have sites outside the city in Duval, near Carnation, and Woodinville as well. So we're really expanding. We expanded our study sites for 2019 and 2020. I actually just wrapped up my 2020 sites before I left for Minnesota. So hopefully we'll be able to get that data in terms of genetic sequencing submitted soon. We did, of course, had COVID delays. I think every scientist worldwide is having delays because of the pandemic. And I actually was really lucky to be able to still have my field season because it had started before the pandemic hit, at least in Seattle. So I feel really, really lucky and really privileged to be able to even have this data, let alone be able to analyze it for next year. But a lot of my work looks at this nutritional analysis element. We're looking to do more of that through a lab at the University of Washington. We are looking at the development and the fitness, or the relative success and survival of the bees in all of those sites. There's a lot of entomological pieces to that that are going to be really exciting to understand, how does what they eat impact how well they do on the landscape, how well their offspring do the next year. And then we also look at weather. The Pacific Northwest, it rains all the time, and the bees don't fly well in the rain. So, we have all these questions of, well, if it rains a lot, or it rains every other day, or the temperature falls in the middle of the season, how does that impact the bees? And we have enough data at this point to kind of take a glance into that and do some statistical analysis of that. And hopefully find some more information. And then we'll do the same sequencing that we did for 2018, for 2019 and 2020, hopefully in the next couple of months, barring any more pandemic delays. And we also analyze for bacteria. That, again, is a whole nother talk in and of itself, but bacteria are responsible for a lot of the development in insects, and we can actually sequence for identifying the bacteria alongside the plants. So we can understand what bacteria might be beneficial to the bees, and even discover new bacteria that are picked up on the surface of flowers. So much more to this than just 2018, but luckily I got to share some of it with you. And with that, I just want a quick thank you, of course, to all of my list of acknowledgements and my advisor, but again, big thank you to Joey and Twee. This is, I kind of like the virtual element. It allows anyone, anywhere to just log in and be a part of the sharing of information. So it was really cool to do this. Of course, I'm always happy to do it again. And I think I hand it over to Twee. I'm happy to take any questions and all questions up until I get hungry enough that I go and eat dinner. So thank you so much for joining us.
Awesome. Thank you so much, Lila. That was phenomenal. I'm going to start pulling questions from the audience now, and everyone feel free to continue asking questions in the chat. But our first question is from Karen. If climate change causes an early spring, why doesn't the warmer weather also cause the bees to wake up earlier?
That's a fantastic question. I didn't include it because I was a little nervous about my ability to talk about it, but hopefully this is clear. So it's a great question. So if the flowers are opening sooner and it's warm, let's say in February 1st, it's like 70 degrees, just crazy, which might not be so crazy soon, but crazy. Why aren't the bees coming out earlier too? So insects develop very differently than plants. Plants essentially need two indicators to start their bloom. That's light. They need a certain amount of light, and during the winter there's usually less because both the sun is further from the earth, or wherever you are at the earth at that point, as well as it's usually cloudy and darker, and they also need heat or temperature change. So warmth and light is going to lead to flowering if it's January or if it's July. Bees, on the other hand, along with most other insects, if not all other insects, work on something called degree days. So if you think of it as like a bee can only continue developing between a very small range of temperatures. So like, let's say you'd need, and I'm making this up because I'm just a mason bee specialist, not an all bee specialist. Let's say a bee needs 75 degrees over 200 degree days to develop fully into an adult bee. Let's pretend that that's the perfect amount of time. And let's pretend that that's the amount of time in the winter. If the temperature goes above 75 degrees or below 75 degrees, that development stops. So it's almost like if in the winter you could only start moving and shoveling your snow or driving your car going to work if it were exactly 35 degrees. And if it were 30 degrees, you couldn't move and your development was halted. And if it were 39 degrees, your development was halted, you couldn't move. You couldn't go drive your car or shovel your snow. And so because of that, it means that bees have a very specific length of time that they need, and it's not influenced by if it's warm or earlier, it's influenced by an exact temperature range. There actually can be detriments to development if it's too hot or it's too cold. That can actually slow down development. It'll never speed it up though. So you get the same timeline for emergence no matter what. That is to say, I place my bees out in March, and usually it needs to be 40 degrees, 50 degrees, usually sustained 50s for them to come out. they could come out if it were 70 degrees, but they won't be ready to come out or able to come out until it's at least 50. And they won't be able to come out until that exact point in time, that exact amount of degree days has passed. It's a lot. So I hope I did a relatively okay job at explaining it, but it's one of those mismatches that's going to become clearer and clearer, I think, over the next couple of years.
Awesome. Thank you for that answer. That's really cool. So the next question is from Trina. What kind of bee house would you suggest putting in the garden at this time of year? And they are from the San Francisco Bay Area. Would you happen to know?
That's helpful. I needed the location because Minnesota is going to be pretty different. That's a good question. I'm a big fan of making your own because I discovered it was very easy. It really is so easy. Obviously, it's a little harder in pandemic times, but I usually try to push people away from buying a house. And this isn't an assumption that you've bought one, but Costco was selling these bee houses, like native bee nesting houses a couple years ago. And they were essentially like bamboo tubes, just like I used, stuck into a house. And they were essentially glued into the house. And there was some room at the top that was kind of open. What we see a lot, at least in Washington, and I would say in Minnesota too, which I'm familiar with pollinator-wise, wasps love to nest in little open areas. You know, you see them at the top of your garage. There's one on the garage of this house right now, and in my hometown as well. There's one right at the very peak of our garage. Wasps love to put their nests like right in that open space. And if there's a food source right below them, like even better, they can just go down and eat the bees when they're bored. So that's not to say that's bad. You know, predators are a natural part of life, but I think that the houses that don't have something stuck in and don't have any open space for wasps to nest in do a lot better. Again, a lot of people clean them out over the winter. What we do see, and it's been pretty well studied at this point, is if you leave a bee nesting house out for years and years and years, it develops a lot of bacteria and viruses that collect, especially in the West Coast, because it's so wet and so moist all the time, that all those viruses and bacteria just kind of live and grow there. And that detracts the bees, and it can also provide an avenue for the viruses and bacteria to get into the bees as well. So it's good to clean them year after year. So you want something you can clean, which PVC pipe's super easy to clean, something you can take apart, those nesting tubes. You don't have to freeze and kill all the bees. You can, you know, put them in your garage and just let them do their thing, and then throw them out in the yard in an area the next year, and the bees will emerge. But at the very least, something you can take in, something you can clean, and something you can put back out. So I would say there's a lot of really cool videos of building your own, but don't feel like you have to listen to all of there's a lot of bee nerds out there who will tell you that, like, you know, plastic doesn't work, you know, PVC is a bad idea, or you have to paint your box black to warm it up. My boxes are so cheap and they work great. So don't overthink it. Just follow kind of those like general guidelines, and you can purchase a lot of the paper materials too online.
Sweet. I feel like I can definitely go down a rabbit hole of like trying to figure out what the best bee house for me would be.
Yeah. Yeah. Don't overthink it.
Awesome. And we have a couple of more questions about houses specifically. What I do with my bee house in the fall is putting it in the garage where it will freeze, best for the bees next year.
There's a very good chance that question's from my dad, because he was asking me the exact same thing the other day. So I'm not super familiar with any mason bee activity in the Midwest and how they deal with it. Honeybees, I don't know if this is interesting to anyone, but honeybees keep themselves warm in the winter by vibrating. Joey and I were kind of talking about that earlier with the hornets, but the honeybees are able to keep warm in that hive setting, but solitary bees, they just exist in their little nesting spot for the whole year. So I think what we do in our lab is, and Seattle does get cold enough to freeze, so I guess this is applicable. We bring them in and we put them in a very fancy growth chamber that I would not expect anyone else to purchase or afford. I could not afford it, but we're able to control for the correct temperature range. So like a just above freezing, usually like 39 degrees. I would think that if those bees were naturally occurring, so if you put out a nesting box and you don't put out bees and you get bees nesting there, those bees have survived the winter, or their mothers have survived the winter, or their mothers before them, right? So somehow they survived the winter, and if they liked that spot, either they were the bad batch of bees and they weren't going to make it no matter what, or they were probably going to do okay there for the winter. So I would say you can throw it in your garage as long as you didn't purchase them from somewhere else. A lot of bee purchasing programs have you send bees back to them in the winter so that they can open them up, take care of them, and put them in these growth chambers or keep them in warm temperatures. I'm not a big fan of the programs that ship bees to you anywhere in the world and then you ship them back, because you're transporting a species that really doesn't belong where you are to a completely new area. You should be able to have enough bees, and you know, we have to improve our conservation efforts, of course, but hopefully there are bees around you that you can admire and have nests in your little nesting boxes, and you don't have to buy them from someone who's not necessarily taking care of them to keep any escapees from getting out into the environment. That's something like, you know, the non-native honeybees are transported all around and they pass diseases really easily to our natives. What's to say that a bee from Seattle can't pass it to a bee in Minnesota? So one thing I think to keep in mind is, like, if your bees are already nesting there, they'll probably do great over the winter. You might not even need to touch the box, but I think throwing it in the garage should be fine. They really should be fine. A lot of people keep them in their fridge. It's like 39 degrees or so in the fridge, but they might dry out. So a lot of information online there too. There's a couple wormholes you can go down.
Definitely. I'm glad you brought up diseases, because one of our questions from Sydney is, are mason bees subject to the same diseases or colony collapse that honeybees suffer from? If not, what are the main threats to mason bee populations in Western Washington?
Yeah, great question. Everyone's very informed. I'm very impressed. So colony collapse disorder, tiny background, I'm sure most of you are familiar, I'm sure all of you have heard of it. Colony collapse disorder is kind of this like myriad of pieces that have led to a decline in honeybees. There are some stats, like honeybee decline stats. We actually have lost more honeybees over the last couple of years than ever, but we've seen higher numbers of beekeeping. And so there's some interesting things alongside honeybee decline specifically and factors that only affect honeybees. But most of the things that are causing decline are things like pesticide usage, chemicals of any sort, a lack of flowers, lack of food sources, a lack of nesting sites due to development. When you pave paradise to put up a parking lot, right, you lose all of the wonderful paradise nesting spots for the bees. As well as things like viruses and pathogens that are passed from non-native species to natives, or are just running rampant right now because of a lot of inbreeding in the bees, because the populations are dropping. So there's all these different factors. They are most definitely affecting all native bees, all solitary species, and solitary bees may be more susceptible to some of that. Social bees like bumblebees and some of the sweat bees are actually social. They have a little bit of defense against that just due to the large population number. You know, if you lose one worker of a bumblebee colony, you're not losing everyone. But if you lose one solitary bee, not only do you lose all the offspring that were inside of her, but all of her children who are now relying on her to bring back food are stuck out in the cold, right? So they're definitely affecting mason bees as well as all of the solitary species in the United States and worldwide. And I think all the things you do to help the honeybees are going to help the native bees. It's, you know, we should think about the worst in our society, help the people who are at the bottom, because it's going to help everyone. So helping the native bees is also going to help the honeybees, and it makes you a little cooler if you're only helping the natives.
I love your analogy for that. Help the people at the bottom first, helps everybody else. Amelia asks, are there any types of beneficial wasps that lay their eggs in mason bee nests, or are they all parasitic?
Beneficial versus parasitic, that's a good question. I'll say this, if you are seeing wasps in your nest and they're not the big yellow scary kind, I guess you could call it, the classic wasp look, if they're a little smaller, maybe they're a little interesting looking, they're not necessarily the big yellow and black striped wasps that we're used to seeing, like dive bombing our picnics at the end of the summer. If you're seeing something other than those, they are most likely parasitizing the bees, but I don't necessarily see parasitism as a bad thing. Actually, as part of our research, so I showed you that we took pollen, and I didn't introduce this other thing, which was that we also take bees and don't kill them to steal their pollen. We allow them to develop fully, we overwinter them in our growth chamber, and we let them emerge the following year. And we actually count how many of those bees have been parasitized, because in a perfect conservation, in a perfect plant pollinator network, of course you have parasitism happening. Of course you have predators happening. You know, the same way we can't just have the mice without the birds eating them. It all has to work in tandem. And so parasitism is not a bad thing, and we use that as a data point. Some areas may be better for parasites than others, or for parasitism, for these predatory wasps than others. But what we see is, when we see parasitism, we see something coming out of the mason bee cocoon or pupa rather than a mason bee. And the development time is usually different. So you see the bees come out first, and then these pupa, which seem like the bees didn't make it, all of a sudden they emerge and there's tons of little wasps that come out. You know, it's something like that. It's very alien-ish if you're not familiar, if you're not really comfortable with entomology. And I don't see it as a bad thing. I think even, you know, yellow jackets eating bees is still a part of life, and it's something to deal with. The only thing is if your nesting materials, if whatever house you put up is just not, it doesn't have the right defense system, it's too easy for the wasps to get in, unless you put it right next to a wasp nest. Of course, that's going to lead to a, you know, higher death rate in your bees that you probably could have prevented. But I just don't see parasitism as a bad thing. I think it's really interesting, and you can look up online the parasitism that kind of happens in mason bees, and they've identified them down to species, know exactly where they came from, and you can track where they've been. It'd make a cool research project. But yeah, I think it's not a bad thing. I'd feel fine about it, unless it's happening to all the bees, and then it's just another interesting data point to me.
Yeah, I'm really glad you brought up the importance of parasitism in like a really good ecosystem as well. You kind of need these varying relationships and different relationships for the ecosystem to function properly.
Right.
Amelia asks, if female mason bees are laid first in the nesting tube, what makes the males hatch first? Do the males just need less time in cocoons?
Oh, good question. Okay, so the males have essentially one duty, and that is to emerge in the kind of early spring, so before the females, and mate with the males. That's their whole job. So after they mate, they essentially kind of kill time until they die. They hang out around the nest. They kind of cause trouble. You know, they just sit there and take up space, and then they die off early. And the females go on to lay all of the offspring, collect all the food, and live a lot longer. So females are living 4 to 6 weeks. Males are living 2 to maybe 4 weeks if they can find enough, you know, food resources to keep them going. But all of that genetic material that's inside the female body, and all of that time that she needs to be alive contributes to her larger size. She's much larger than the males. And it also means it takes longer for her to develop. And so there's an idea that the female bee takes more nutritious pollen. You know, if your female is going to do all the work and your male is just going to die off after a week, you should probably feed the female offspring a much higher amount of really, really good quality pollen. So a lot of the times the bees are set up in a way where they lay the offspring of the bees are able to do this. Forgive me, my advisor would kill me, but I forget the word for exactly how they lay a male versus a female offspring, but they will lay the male first in the nest and the female last. And so I think that a lot of people will see the males coming out and think, oh, my nest isn't doing very well. But in reality, the females are coming, they just come later, and they kind of crawl through where the males used to be and out into the world. So it does take longer for them to develop, so they do have a lot more to do in the world because of it.
This is my own personal question, but I'm a horticulture student, and I really loved your breakdown of plants, and I got really excited when you talked about the different genuses. But is there any research at all on the differences in diets between males and females? Because you mentioned that females would need a higher nutritional value diet.
Yeah, I'm kind of digging into that literature now. It's a perfect question. That's part of one of the pieces we're studying. So one of the chapters of my dissertation is looking at the breakdown of sex ratios, male to female sex ratio, and the different foraging patterns for male versus female bees. And because it's nice I described that system of males in the front, females in the back, because that's kind of a known system for how the bees nest in a human nesting style, you know, where I'm putting out a nest structure, they're not trying to find it in the wild. Because we know that's how they tend to nest, we can actually take pollen from the front and pollen from the back and compare them. And so 2019, we started doing that. And sadly, I don't have the data because of COVID and personal PhD delays that happen all the time. And so that'll be something we know by hopefully this time next year, hopefully a lot earlier this time next year. But we can look at, you know, do their foraging patterns change? Is the nutritional quality different? I would assume yes, but I actually kind of think I'm wrong. Just seeing how the bees do where they go on the landscape and how little sense it seems to make, how little order there seems to be. I almost think we won't see it, but I'm really interested to get the nutritional data back, because we'll send it off to a lab that does like a really intense breakdown of the nutrients, the amino acids, every single amino acid, as well as the lipids, the sterols or fats. So to be determined, but my guess is there's assumed to be a difference, and there probably isn't, because I'm constantly proved wrong by these bees.
And in the lab, do you use the PCR method to kind of differentiate the different pollen species?
We do some, we can. So what PCR allows you to identify if you know what you're looking for. But because most of the time we don't know what we're looking for, we end up sending it off to a much fancier machine that uses, it's essentially like PCR on steroids, where they don't necessarily need a reference, they just do all of it. And you can do a mixed sample, which is, we essentially have to use this fancy software because it's a mixed sample. If we had just one type of pollen, if we just went out to a willow tree and took a little brush and took some pollen, we could do it all in my lab at UW. But because we don't have the infrastructure to do these mixed pollen samples, where you have to barcode essentially every single pollen sequence, every single piece of DNA, it becomes a lot harder. So we end up sending it off. So that's why the delay is. If I could do it, I would just be up all night for a week doing them all at once, you know. A little bit desperate to get some data, but sadly we'll have to send it off to a lab actually in Nova Scotia, so in Canada.
Yeah, pandemic makes doing research really hard. Sue asks, what is a good resource for attracting more mason bees to my urban yard and building mason bee houses? So touching on what we talked previously. Yeah,
I mean, mason bee houses are definitely a pretty easy build. You really just need a wood case and a bunch of tubes. They'll do great with that. Bees love sun, so if you have a sunny area, I think planting a lot of pollinator-friendly plants in that area is always good for attracting bees. Also, I've noticed this cropping up a lot more. Leaving a lot of your land unmowed or unkept is really great for bees. It looks terrible in terms of our like city ordinances, but it's actually fantastic for nesting sites. So you get a lot more bees nesting in like leaf litter that you leave on the ground. If you don't rake all the leaves in the fall, you just let them be, and then you let the snow fall on top of them, at least in the Midwest, where snow falls more often than in Seattle. But on the east side or on the west side of the mountains, just not in the city, you could do that too. And that gives a lot of nesting opportunity, which means that there will be more bees the next year. So, you know, whatever you do this year, your efforts this year will hopefully be rewarded the following year. If you plant plants this year, you should see the fruits of your labor the next year in terms of bees. But flowers are always just great. A lot of those pollinator-friendly gardens too are like, you plant it and forget about it. You never have to touch it. You know, you don't have to mow it, you don't have to pull it, you don't have to weed it. It just kind of goes because it's like a natural landscape. So I think those are beautiful. There's a lot in the city in Seattle, and even where I am now, there's lots of those. So I think that's probably number one. And then number two is maybe some nesting sites for the next year.
Sweet. I'm going to plug native plants for a bit here. Yeah, Lila included a ton of different genuses that you can kind of look into. And yeah, the maintenance on them is less compared to ornamental or invasive non-native species. Patricia was the person who asked about what to do with these bees during the winter. And they asked, should I put the house during winter in an airtight plastic bag?
Oh, it's an interesting question. Well, I would say no. These are kind of funny how they breathe. Insects don't breathe the way that we breathe. They also don't need water the way we need water. It's all very different. I wouldn't seal it because you don't want to accidentally suffocate them. Know that they use like a fraction of the air that you think they use. I keep them in sealed containers, but I poke a couple holes in them. If you're worried about like wasps getting out, which I know a lot of people are, even if the wasp went in and then you close up the bag, and if you were to open the bag, the wasp would get out type of thing, I think you could put it in like a Tupperware container or one of those big plastic containers, like gallon or couple gallon containers, and then put a top on it, and you could just poke some holes in the container. That would be fine. That'll kind of solve the problem of like, I don't want a bunch of, what if there was an ant that got it? You know, I don't want a bunch of bugs in my house or in my garage. I totally get that. I think a plastic bag might be too tight, but throw them into your garage in a container of some sort, and they should be fine.
And they also ask, how do you make those paper tubes?
Oh, I do not make them. Great question. I get them from a guy because I'm a PhD student. I can just get them from a guy. They do sell them, and there are a lot of paper companies that actually sell them. They use them for all kinds of things. They don't make them just for bees. So it's kind of a sneaky trick, but like lots of companies sell them for these exorbitant prices that you should not be paying more than a couple cents per tube in reality. And you can order a bunch. So if you really like mason bee keeping and you want to just order a bunch and share them with your friends, you can bulk order from a lot of these paper companies for really, really cheap. And of course it supports the paper company and also provides you with a bunch of cheap tubes. And I slit mine down the middle with a paper cutter. I actually spent hours in the lab just slitting tubes in the fall and in the winter of last year and the year before. I think I split about 4,000 tubes, and it's kind of fun. I know a guy who does it in front of the football games. He just slits tubes in the football games. He becomes mindless and easy. So you can do that, but that's only if you want to open them up to check inside. You don't have to do that if you're just keeping them by yourself. You can just wait until the bees have gotten into their cocoons, and then you can either open up the tubes or just leave them to emerge straight out of the tube the next year. So I think that's the best idea. I don't love the idea of like capitalism controlling whether or not you raise bees. You know, like if you're gonna buy them online and it's like $40 for a kit to buy them online, you can make them a lot cheaper. Like you don't have to do that. I'm telling you, there's another way. And those paper companies are really cool. So there's a lot of options there.
Amelia also asked, do you harvest the cocoons of your mason bees, and would you recommend it?
So I do, because we use the cocoons that the mason bees produce so the mason bees for 2019 laid a bunch of offspring. Those offspring developed into pupa or cocoons. They're technically, etymologically, they're pupa, but I think cocoon really sums up what most people understand cocoon more than they understand pupa. It gets into the weeds, it's not worth it. So they produce all these cocoons that house their offspring. We put those in a growth chamber and keep them at winter temperatures and then allow them to emerge the following year. So they're really important for us because that tells us, you know, how many offspring the bees laid, as well as how many of those offspring made it through the winter, because making it through the winter is necessary for survival, and you can only do that if you have the right fat storages in your body. So that points to a nutrient quality thing. Are they getting enough fats in their diet to be able to survive the winter? And then it's also important to know, once they do emerge, how many are females versus males? You know, what are the ratios of females to males that we're getting? Because really high ratios of male to female is very bad. In terms of inbreeding, it'll cause a lot of issues. When you don't have a lot of females, it usually causes the population to fall into decline really quickly. And so what you want is an equal number of males to females. And so that's another data point that we can look at to say, okay, well, this site, there were 40 males and 2 females. So that tells us something. Maybe it tells us something about the nutrient quality of the pollen, or what they're foraging on, or pesticides or chemical, something about the bees versus a site where it's 50-50. So I do. I don't think you have to. People do because they clean them off. They'll clean the cocoons off. There's lots of videos online, really beautiful videos of people doing that. And anyone, of course, can do that. It is really fun in the fall to like open up all your tubes and look at all your cocoons. You could find the ones that are parasitized because you can actually see in the light what they look like. They're a little lighter in color, and you can see through them a lot. You can see how many did make it. And you can see exactly how many offspring your bees laid. And so you can get an idea of like, okay, well, if I put out this nest and I got, I didn't put anything out with it, just the nest, and I got 100 bees out of it. That must mean there are bees in my area. So it's kind of fun. There's nothing wrong with it. There's also nothing wrong with leaving the box in your garage and then just putting it out the next year and letting them all come out naturally. So either way, it's kind of a personal preference thing, but kids love it too. You can like hold a bee in your hand. It's kind of cool.
That is very cool. Ooh. I think we only have time. It is 7:15, so we might only have time for like one last question. But Rachel Lodge asks, can you talk about how bees use nectar as opposed to pollen? And do your mason bees collect nectar? That might be a very loaded question, but.
No, it's great. That's a great question because I didn't talk about nectar much. I really talked about pollen, so good eye. So bees do use nectar. It's probably the predominant food source that they're picking up when they're flying around. When you see a bee visiting a flower, they're going for nectar. They're not going for pollen. Pollen is kind of a collected, when the bee tunnels into a flower, they end up passing through all of the anthers of the flower that hold all that pollen. So the pollen just coats their body whether they want it to or not. That picture with the pollen one where I had this bee essentially covered in pollen from head to toe or from head to thorax is pretty standard for how the bees end up picking up all this pollen. And so, of course, the bees are collecting nectar. The nectar is used for energy in flight. So nectar is just basic sugars. If you ever have tasted like honeysuckle, that's nectar. It tastes great to us. And it's what makes honey taste so good, kind of the sweetener to honey. And nectar provides all of this energy, especially for bees who are flying for, all hours of the day collecting pollen to bring back to the nest. So it's definitely super, super important, and it's something that they pick up. Some bees pick up pollen by eating it, and they end up kind of like vomiting up. It's very cute. It's like sugar water. They've never eaten anything other than pollen and sugar. So it ends up being kind of this mixture of like pollen and nectar compacted together. And we do see nectar in the pollen balls, and that picture of pollen balls. The reason they're so kind of cakey and sometimes really squishy depends on the amount of nectar that's been mixed in, either through this kind of like vomiting process or through just, mingling in the flower with the nectar itself when it touches the bee, and the bee rubs through a bunch of nectar and it gets all over the place. So it's part of it. A lot of research has showed that it doesn't make a huge impact in how the bees do, whether there's a lot of nectar in that pollen provision, that sample, or not. It more adds to the ability of that pollen ball to stay together, so that the larva isn't like searching all over the place to find the ball of pollen or the loose pollen everywhere. And I mean, you can think of it as like, you see pollen blowing through the air, that's going to be pretty hard for a bee to pick up versus this tight little nice ball that they can feed on. So it's part of my studies. I think it's such simple sugars that it's really not as important as the proteins and the fats. So it's not something we study, but it's definitely a topic of study. It was a great question.
I really love the cute bee throwing up part. Very true. All right, folks, we are running out of time. So thank you, Lila, for sharing your amazing research on mason bees with us tonight. Next week, J.R. Harris will be sharing stories of backpacking and adventure from his book, Way Out There. You can find details for this and other online presentations on our website. Thank you, everyone, for spending their evening with us. We hope to see your names at the next event. My name is Twee, and have a wonderful night.
Thanks, everyone. Thanks, Twee.
Thank you, Lila.